Morning. Very happy to see you, sir.
Same here, madam. Am I audible to you?
Yes, sir.
All right.
Your image, I think, lot of it's.
There is a blurriness in the image, right?
Yes, exactly, sir.
That's okay. Image doesn't matter. What I talk matters.
That's true, but even our participants would like to see you, is it not, sir?
Yeah, because during the course of my talk, I will keep my video shut because there could be some internet issue. That's the reason why I will keep my video shut, and so that it will be seamless.
Okay, no problem, sir. Maybe I was thinking in Bathinda there is a lot of no sun there.
No, ma'am,
So
we are located in a very rural area, and therefore the connectivity, is a problem here.
okay, sir. No issue, sir. So, yes, dear participants, it is already 10 o'clock. We'll start for today's session. I welcome our resource person today. He is none other than very dynamic Dr. Chaitanya from Punjab Central University, Department of Zoology. I am really very happy to inform that he is one of my very old friend during my PhD days at Hyderabad Central University. Sir was working with our mother, we can say that Dr. Aparna Datta Gupta. So he was a pet to madam, I can say. So so that was my association, and today I'm very proud that sir is at Central University of Punjab. He is also belonging to my entomology. So this is the short what is my association with Dr. Chaitanya. And now let me give a brief introduction of Dr. Krishna Chaitanya. He is actually, his research objectives is to elucidate the pest resistance in transgenic cotton, to decipher early events of plasmodium evasion in the malaria mosquito, to investigate the thermoplastic degrading potential of the insect gut microflora, and to establish the Drosophila model system to investigate the obesity related disorders. Sir has the peer recognition in so many members. He's a member for Entomological Society of India. He's adjunct professor for Centurion University of Technology and Management, Odisha. Zoology question paper setter for CUSET PG conducted by NTA. Life member for Association of Entomologists, Punjab University, Patiala. DST Serb Young Scientist India. CSAR Research Association India. SRF, JRF, and he's also NET qualified in the year 2005. So he has completed many research grants, more than 85 lakhs that are funded from SERP funded co-research grant and UGC funded, Central University of Punjab funded, and there are many more grants that has been sanctioned by ICMR DHR schemes, and he has got research publications with an impact factor of 71.15 and total citations is 340. You can see his potential, so much of this thing he has towards inclination towards research. He has got many publications, more than 50 publications and 10 book chapters. He's ongoing so many PhD students and dissertations he has for MSc 20, and ongoing are 5 PhD students. He has given lot of invited lectures at various platforms. He is a keynote speaker for the national seminar on impact of chemicals and drugs on COVID-19 at Guntur, Andhra Pradesh, and also at different parts of South India like Kerala and Patiala in the north. He was also a member organizing committee member for various national and international conferences. He has, he has done so many skill development programs, especially on the development of EPG Patashala module that is actually need of our my dear participants, which we have to learn as a teachers. So with this, there are many more with this small this thing. I am really very happy, sir, for accepting our invitation for my participants. Let me give you a small introduction about my participants. They're all from the different parts of India, especially like, Tamil Nadu, Central University, Maharashtra, Andhra Pradesh, and Telangana. They're all UG and PG teachers from life sciences. This is actually an online, it is a UGC Academic Staff College, HRDC It is a refresher course in life sciences for the UG and PG teachers, which is mandatory for them to in order to get promotions. So there are near about 37 to 39 participants. So this is they're from botany, zoology, microbiology, biotechnology, biochemistry, genetics. So all are from life sciences, sir. So now over to Dr. Krishna Chaitanya.
Am I audible to you, madam?
Yes, sir.
All right. That was a very lengthy and kind introduction from you. Um, as you are saying, your participants are all UG, PG level teachers. So I really don't know what I should teach them. I should actually learn from them. But given the mandate of, UGC HRDC refresher course, uh, I choose to speak on genetically modified crops. The reason why I chose this topic is because I thought it will cater to most of the participants because they are from the background of botany, zoology, microbiology, and biochemistry. So, during the next 1 hour or 1 hour 15 minutes, I'll be speaking about genetically modified crops, and I will switch off the video because of certain internet issues, and I shall open my presentation. So, after 1 hour 15 minutes, I'll be very glad to take up any kind of questions or queries from the participants. Is the slide visible, madam?
Yes, sir. It is
Can
visible.
you can
Yes,
you read
sir.
the slide?
Yes, yes.
Yes.
Okay. Well, once again, thanks a lot, Dr. Madhavi Garu, and I welcome all the participants for this session. Uh, my title is genetically modified crops, a bird's eye view. So I would like to, in the next 1 hour, divide my talk into 4 parts. Initially, I'll talk about genetically modified crops, the history behind it, the global status, and the advantages associated with it. The second part of my talk covers how a crop can be genetically modified, what are the various steps involved in that. The third part of my talk would include particularly about Indian transgenic crops, that is the story of Indian transgenic cotton, as well as uh, the latest approved genetically modified mustard. And at the end, I would like to talk about the perils associated with the GM crops and the upcoming new technologies with respect to transgenics in India as well as abroad. I picked up this slide from a global, relevantly website, where it speaks about the concern of food security. So, if you can see, we are having ever expanding population, and somewhere we are projecting this population would be around 9 plus billion in 2050 and 10 plus billion in around 2100. And it is our prime importance, it is of prime importance to actually feed all of these population, and the available land area which we have, arable land means the land which is cultivable. So arable land you have is 1.5 billion hectares globally, and you have almost, you say, 2050 you have 9 plus billion population, and 2100 you have this much population. If you divide this arable land with the amount of population you have, so it is roughly around, zero point 0.1 to zero point 0.2 hectares per person, which is required for his or her survival. So, but actually, what we require, But what we actually require is zero point 0.5 hectare per person. But what we are going to get in the future is not even zero point 0.2 hectares per person. This is like, one person to be fed for an year. So that means we have a food security, which is a formidable challenge, not only in India and but also abroad as well as globally. Now you see, when you talk about population again, in 2050, this is a slide which projects for 2050, and you see that, where do the hungry live? The most of the people who require food would actually live in Asia and Pacific, that is, India, Pakistan, and these regions. And you see that approximately 578 million people are falling under Asia and Pacific. And you see, this is, I'm trying to, make a context here. I mean, like, I'm trying to set a context is why, we need enhanced food productivity, why we need enhanced technologies for providing higher yields of food as well as nutritious food in both ways. So when you talk about India particularly, the population is rising, and the arable land is, when you divide this arable land versus the population currently, this is the current state of statistics, and you see that at this point of time we have zero point 0.1 hectare per person available, uh, for which you need to produce the food. But actually, ideally, we need zero point 0.5 hectares. That means the space available to us is very, very less, but the food demand is absolutely very high. So, therefore, from the last three slides, what we can infer is, food productivity has to be enhanced. So, what were the steps taken in that direction? So the first one was, conventional breeding. So before genetically modified crops came into existence, or even before transgenic crops came into existence, there were practices of conventional breeding wherein you choose two varieties of plants. I'm putting it in a very layman language. I'm not going into the deeper technicalities of it. Wherein you choose two different plant varieties which are closely related. Closely related means they belong to either same species or closely related species. And you pick up two varieties which with the different properties. Like, say for example, one is a high yielding variety and one is a drought tolerant variety, or one is a dwarf variety and one is a what do you say, stress tolerant variety. So you actually, or disease tolerant variety. So you actually try to form hybrids among these varieties. So initially, the practice was of conventional breedings, and we actually achieved success to some extent with respect to this. Like, for example, if you see this, you have various crop varieties that are resistant to diseases. Like, in, for example, wheat, we have himagiri, and cowpea, chili, cauliflower. You have various disease resistant varieties which are produced by conventional breeding. They are resistant to certain diseases, like white rust, black rot, bacterial blight. So again, when you talk about certain varieties which are resistant to pests, so you have various, okra. Okra is nothing but bhindi, and it is generally, infested by Helicoverpa and Spodoptera. So you have brassica, rapeseed mustard, flat bean. You have various varieties available which are developed by conventional breeding. That is, which provide resistance from pests. And again, you have high yielding varieties of plants. Like, for example, classic example we have is of rice, IR8 variety, Jaya, Ratna, Sonalika in wheat, Kalyan Sona, and there are so many disease-resistant and high-yield varieties which are obtained by conventional breeding. And the target for all these things majorly is threefold. That is, one, to obtain a high yield variety of a plant or a crop, and one, it has to be disease resistant. Three, it has to be pest resistant. Now, these are the three aims with which we were able to, or I tried to perform conventional breeding, and we obtained success to some extent initially. But there are certain limitations associated with conventional breeding. That is, the first and foremost thing is when you are breeding an X plant with a Y plant variety, you are not actually targeting a particular gene, you are actually targeting the whole gene pool of both plant varieties. So that means that the genes which you want, you will get it in F1 hybrid, but at the same time you will also get certain undesirable genes that can be transferred along with the desirable genes. So that would be probably detrimental. Like for example, you have developed a high yield variety of a plant using conventional breeding, and you you find that this particular variety needs a lot of mineral or a lot of water, or it is it requires certain specific conditions to grow. So in that sense, conventional breeding becomes disadvantageous. And during the conventional breeding, if to obtain the desired variety, you need to at least cross for 14 to 15 generations to get a desired trait. The desired trait will be at least 10 to 15 generations down the lane, you will get the desired trait. So that means the time taken to achieve your desired result is higher, and you don't have any control over certain genetical phenomena like genetic recombination in case of conventional breeding. And the foremost thing is it is limited to exchanges between same or very closely related species. That means you can take a plant X plant and a Y plant with different traits and which are closely related, and you can make an F1 hybrid. Like say for example, if you have some other property, like in case of bacteria, in case of bacteria, you have a different property which you want to put it in a plant. You cannot do this with respect to conventional breeding. So having said that, okay, you have the issue of food security, which is perennial, which is global, and the initial technique which was used to develop high yield varieties was conventional breeding. Now, the another strategy which was used, to increase higher yields or to get higher yields was excessive usage of insecticides and pesticides. Um, I think many of you are aware of this fact that DDT came into picture in 1920s or 1930s, which was approved then, and the scientist called Muller obtained a Nobel Prize for that as well. But unfortunately, excessive and indiscriminate use of this DDT definitely gave higher yields initially, but unfortunately, this DDT had many side effects and many environmental hazards associated with it. And down the lane, something which was considered to be most successful, and for which for that finding somebody has received a Nobel Prize, and down the lane after 50 to 60 years, that particular DDT was banned entirely. So, likewise, you see that, the DDT, which was entering into the environment to water contamination, is getting accumulated, which we call the process as biomagnification, into smaller organisms to higher organisms. And we have seen that during that period, many birds have got extinct because of the effects of DDT. Not only DDT, actually, there are so many organochlorines, there are so many, pesticides, and there are so many insecticides which have caused a lot of environmental hazards due course of time, which were banned. And now that means now you should have an alternative. And slowly, when you are using a lot of insecticides and pesticides, now the pests and insects have got resistant to those insecticides and pesticides uh, because of excessive usage and the process of natural selection. Now that means when you have excessive use of pesticides and insecticides, you probably have got higher yields to some extent. But again, if that is causing serious health hazards to human consumption, particularly, and it is also generating more amount of pests, and in a way, these pests are destroying the crops. So we or the scientists started to think about an alternative, and the alternative was genetically modified crops. So we had an era where we have used conventional breeding and we obtained success to some extent, but it has its own limitations. And we also have an era where we have been excessively using insecticides and pesticides, and then we have we wanted to find more alternative as well as sustainable solutions. And therefore here the genetically modified crops came into existence. I hope I am audible to you, madam.
Yes, sir. Yes,
Yes,
sir.
sir. Yes.
Yes, sir. No.
All right.
Yes, sir.
So now we the next part I will be talking about how a genetically, this is the background which I have set. The background is this, and the history associated with that, the what do you say, the initiative of GM crop. Now, what is a GM crop? And if you have to go by the scientific definition of it, which is given by United Nations Cartagena Protocol, and it says that GM it's a living modified organism. A plant is said to be genetically modified if it meets two requirements. One, the plant contains a novel combination of genetic material. This means a plant will have its own genome, but by through some means, if you are able to put some exogenous DNA into this genome, the plant contains a novel combination of genetic material then. And how do you put this genetic material or a gene for that matter into the genome? By using modern biotechnology, or now we call it in various names like genetic engineering, recombinant DNA technology, and all these names. So, these words are used interchangeably. That means GM crop, genetically modified crop. Sometimes we call this as biotech crop. Sometimes we use it as transgenic plants. And sometimes we use these terminologies. And the technology which we are using to produce the GM crop is we tend to say it as biotechnology, recombinant DNA technology, or genetic engineering or gene manipulation for that matter. So then what are the advantages? Genetic engineering or recombinant DNA technology has to have some kind of advantage over conventional breeding, else it is of no use, right? So when you have limitations, we discussed about conventional breeding, and we also discussed the health hazards of various pesticides and insecticides. So genetic engineering of plants provides solution in this regard. It allows the direct transfer of one or just a few genes between either closely or distantly related organisms. The point to be noted here is, one, you can selectively pick a gene and put it in the genome of the plant. Two, it need not be closely related species of plants. It can be distantly related. That means I can pick up a particular gene which is useful from a bacteria or an E. coli, and I can pick up the gene using the technology we have, and we can put it in the plant of interest and get that desired trait in that particular plant. And again, the second advantage we have with respect to GM crop is this, the crop improvement can be achieved in a shorter time compared to conventional breeding. That means you just need 4 or 5 generations to get your desired trait. Then again, you have um, allows plants to be modified by removing or switching off particular genes. This I'll discuss a little bit later um, in the subsequent part of my presentation. So, what advantages we have for if we have biotech crops, and how does it contribute to food security, sustainability, and climate change mitigation? So, you can see here in this slide. First thing is, once the first transgenic plant has has been developed, right, in 1990s, way late back in 1990s or 2000 around. From then to, if you take next 20 years, you see that the crop productivity has increased enormously. Two, it conserves biodiversity, saved 183 million hectares of land from plowing and cultivation. Now, how does this happen? How we help biodiversity is, when you have high yields in a land, in a particular land, you do not look for, explore for lands which are available in the form of forests and wetlands. So therefore, we are in a way saving or conserving biodiversity uh, by adopting transgenic crop. Then again, because you are using this biotechnology, there is a decreased use of insecticides, pesticides, and herbicides compared to 1980s. If you are talking in 2020 terms, then definitely the herbicide and insecticide usage has come down significantly. Then again, obviously, when you are having high yield, that is contributing to the poverty and hunger. That is actually alleviating poverty and hunger. And the fifth important aspect which we have to talk about in terms of GM crops is its reduced CO2 emissions. So when when we are getting a high yield varieties, and when that particular variety is also, like, say, herbicide tolerant, and also that's what happens is, you do not have more amount of weeds in the fields. That is because when you are applying a herbicide tolerant herbicide on the crops, the main crop is always herbicide tolerant, and the herbs are now eliminating all the weeds in the field. And these weeds, like grasses and all, they die and they become a natural form, which are laid upon the soil, and which prevents soil erosion. So that means you are in a conserved practice, you have to till your land. You have to till your land, and this tillage of land will actually release a lot of carbon or carbon dioxide. So this particular phenomenon, where when you have a herbicide tolerant main crop, and the herbicide is effectively eliminating the weed, it is protecting us from the soil erosion, as well as reduced carbon emissions, because you have reduced tillage. Now, let us examine the global status of biotech crops which are grown. So you see that almost over more than 30 countries, in more than 30 countries, we have planted biotech crops since 1996. And as on 2022, the number is approximately 30. And you see various crops are transgenic with respect to various countries. Like in India, the only the approved transgenic crop is, so far, it's transgenic cotton. And recently we have a GAEC approval for transgenic mustard. But you see, in countries like United States, Canada, and other places, you have many crops which are transgenic in nature. So when you talk about the arable area where the transgenic crops are sown, you can see that USA, Brazil, Argentina, and Canada and India. We have these top five. Now India stands at number 5th position at this point of time. But the major players are USA, Brazil, Argentina, Canada, and India, where you have maximum amount of hectares where biotech crops are sown. But you see that India has only opted for cotton, which is not edible. But in case of USA or Brazil, you have so many transgenic crops which are edible as well. So among these 5 countries also, the top 5 grown biotech crops, are biotech soybeans, biotech maize. I'm talking, when I use biotech, that means it is genetically modified soybean. Now, how this is genetically modified, we will discuss this in part 2. Then you have biotech maize, biotech cotton, biotech canola, and biotech alfalfa. These are the major transgenic crops which are available currently across the globe. So to put in a very simple terminology, first key, how we do this, how we generate a transgenic crop. So initially you have to choose a particular gene which is of your interest. Like for example, I will take a particular gene called crygene or crytoxin, which is produced by or which is synthesized by a bacteria known as Bacillus thuringiensis, which is a soil bacteria. Now you have this gene crytoxin, which is which if available in the plant can kill most of the insect pests. Now, how do I transfer my gene? If you have this gene in the bacteria, right, which is a soil bacteria, and now you have to transfer this gene into a plant. You have to put it in a plant. So how do you do that? So first of all, using various molecular biology techniques, you have to isolate this gene from the bacteria. You have to put it in a vacuum chamber where you have
Hello, sir. Sir, you are not audible, sir.
You are not audible, sir.
can I क्या connect कर लीजिए? Hello.
Sir, you are audible, no problem.
Okay, sir.
Yeah, am I audible now?
Yes, sir.
Yes, sir. Yes, sir. Yes,
Yes,
sir.
sir.
Yes, sir. Now you are audible, sir.
Yes, sir. Yes, sir.
Sorry about that. So here I was discussing that we have a gene in the bacteria where we have to transfer this gene into a plant portion. It could be an entire plant or whatever is the requirement, whether it should be in the form, it should be expressed in the leaves, it should be expressed in the flower, it should be expressed elsewhere. And you have to grow these initially in the plant cells. The advantage of plant cells is you can play around with them with respect to their totipotency. That means there are so many ways in which we can grow these plants
You are not audible, sir.
Maybe some internet issues, madam.
Dr. Chaitanya. Is it Batinda, no? some rural area.
Ma'am, not really rural, ma'am.
I went there, yeah, but I can say when compared to Hyderabad and South India,
Yeah, ma'am. We can't compare it with Hyderabad, but yeah,
yeah.
as compared to Hyderabad, yes, of course.
2019 for Indian Science Congress, it was at Punjab. I think many of you know. 19 January, I was I went there for invited talk. So then I could see all those areas. That was the first time before COVID. 19 January, Indian Science Congress at Jalandhar, lovely professional university.
Yeah,
So
ma'am. Yeah.
yeah, it was totally remote. I was really shocked. I was thinking, like from my childhood, North India, bole to kitta acha rehta. What is this?
Yeah, ma'am. Not every place will be developed, so it's like that. But lovely professional university. I think they have got nice campus.
Yeah, beautiful. It is.
Uh,
LPU is really like London, I can say, UK.
yeah, ma'am. They have got very nice campus.
Yeah, but the way that is approaching to that university and all. Sir,
Ma'am, I think sir left the meeting.
no,
Chaitanya
no,
sir.
he's there. I can see him. Sir, you're unmute. Kindly mute yourself, Dr. Chaitanya. So that was my experience with Punjab. And again, dear participants, this Indian Science Congress this January, I think many of you know, it is at Nagpur University.
Yeah, ma'am.
Mm. Again, I'm going.
Okay, nice, ma'am.
Yeah, we can meet if anybody interested. This is again Deepika. You can come with me, dear.
Deepika, ma'am, your mic is off, ma'am.
Yeah,
Unmute your.
Yes, ma'am. I thought like I'll just call you in that context. I
yeah,
think I'm okay to come.
very good. We'll book the tickets then. I have this habit of just going here and there for this academic this things and all, and I see
Yeah, ma'am.
everywhere, and again I'll conduct here. That is a very I don't know whether it is good or bad, but every year I will do this.
That's nice, ma'am. Take inspiration and then organize it.
Yeah. I'll just call him 1 second. Huh? What is the issue?
Yeah, ma'am.
Okay. Hanji madam, I'm joining. I'm joining. Yeah, I'm joining. Just give me a minute, huh? Share karo. Yeah, hello everyone.
Sir. Okay, sir. It's visible, sir.
Yeah, yeah.
Yeah,
It's visible.
am I audible as well?
Yes,
Yes,
sir. Yes, sir.
Yes, sir. Yes, sir.
All right. So moving ahead. So as I told you, the biggest advantage of genetic engineering is you can pick up any gene from any other source, not only plant, and you have various techniques available through which you can insert it into the plant genome and get the desired result. So most of the transgenic crops which are available or which are grown across the world has majorly, few properties like insect resistance, drought tolerance, uh, and viral disease resistance, insect resistance, drought tolerance. These three are the major and herbicide tolerance. So you have various crops, and the genes we are picking up to insert into the plant are mostly from various bacteria. So who are making these transgenic crops? There are so many players involved, which are both multinational as well as Indian companies. Uh, Certain big names include Bayer's Crop Science. Am I audible to you?
Yes.
Yes,
Yeah.
sir.
Yes,
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 sir.
sir. Yes,
So
sir.
Yes, sir. Yes, sir.
Bayer's Crop Science, Monsanto, Syngenta, and Dow AgriSciences. These are certain multinational companies which are involved in development of transgenic crops. And when it comes to India, we have Central Institute for Cotton Research and University of Agricultural Sciences, Dharwad. We have Maharashtra Hybrid Seed Company. We have Metahelix Life Sciences, Nath Seeds, Global Transgenics. These are certain, uh, you know, big companies which are involved in development of so when you talk about transgenic crops, they talk in terminology called an event. Whenever you are examining any kind of taking seeds from a market or from any company for that matter, they talk about events. Events are nothing but they are like trademark or patented in those companies, like where this particular gene is inserted in your in the plant host genome. So this is the host genome. This is, let us say, the green one is the plant genome, and this is the gene of interest which you have to insert. And for that, this gene of interest should have a particular promoter. Promoter is an upstream regulatory element which is required for transcription and production of protein. And you should have a terminator sequence so that the gene of interest, the gene of interest is starting at this point, and the desired product you will get it at this point. So thereafter there has to be a terminator. So you have a promoter, you have a gene of your interest, you have a terminator, which you now insert it into the host genome. So once you insert it into the host genome, that is the plant genome, you have to examine three things. The first thing is, once you insert, whether your inserted gene is present or not. That means the blue one, whether your inserted gene is present or not. Okay, my inserted gene is present. Number two, if it is present, what is its orientation? Whether it is oriented properly, whether the orientation is reverse, or whether it got truncated. So then you have to examine this promoter sequence and the terminator sequence. All right. Then the third step which I have to examine is, okay, you have your insert and you have its promoter and terminator in a proper orientation. Then we have to examine whether it has properly inserted into the plant genome, whether the directionality and everything else is fine. So if what promoter has been used, okay, gene we know, what terminator sequences we have used, and whether we have made any kind of modifications to these promoter. Sometimes we modify promoter sequences for better expression. So what we do is, that is also like patented in the sense that they won't reveal it, the companies won't reveal it. So you have certain modifications, codon optimizations, which they will take, which they do here for better expression in a setup. You have your gene and where it is getting inserted into the plant genome. Now these things are very important when you talk about transgenic crop. Now there are various steps involved in generation of a GM crop. It's not as easy as we think that we have to just pick up one gene and just put it in a plant genome and that's it, and that plant will express whatever we want. It doesn't. It sounds simple, but it is not. So you have various source organisms like animals, bacteria. So you have picked up a gene from bacteria. You have to isolate its gene at genomic level or at genomic DNA, and then you have to amplify your gene of interest. See, when you are isolating a DNA or RNA from a bacteria, uh, when you isolate a genomic DNA for that matter, uh, you have to again know which gene are you trying to pick and insert it into the plant. So you have to amplify that particular gene. You have to clone that particular gene in a vector system. So vector system is something which you have a promoter, a selectable marker. Okay, so that is, once you have that, you vector is something like a vehicle. So you put your gene into the vehicle, and that vehicle will drive into the region where you actually expect the gene to be inserted into the plant. Then how do you insert, once you have a gene of interest in a vector, that vector has to be placed inside the plant. All right. So plant callus or plant leaf, shoot, meristem, you can grow these plant cells in laboratory conditions, and into these cells you can insert your vector which is containing your gene of interest, and that can be done in two types. That is, one is agrobacterium based, the other is gene gun based. And once you have this agrobacterium and gene gun based methods to insert your vector which holds your gene, which inserts it into the target tissue now, and in the target tissue it has to be expressed. And once it is expressed, uh, the first generation arises. You have to test it. You have to test it for all these three things which I just discussed. That means whether my element is present, whether the alignment and orientation is correct, whether it got inserted at the place which I want it to insert. And then we have to test for the expression. How will we test the expression of that particular protein? Because you have inserted a gene. Now this gene has to give rise to a protein. So when the protein is expressed, you can actually pick up that plant leaf, or you can isolate protein, and you can do certain techniques like Western blotting. Now there are various methods where how we can grow plants under laboratory conditions. Like you can pick up a leaf explant and pick up a somatic cell, make a somatic embryo, and then you can generate a seedling out of it. Or you can pick up an embryo, immature embryo in the form of a seed, embryonic callus. Then again you have somatic embryo. Then again you can generate a plantlet. So there are various methods by which you can develop these plants in the laboratory condition and then shift to the field condition. So but before that, you also along those explant sources, you should also have plant growth regulators such as auxin, gibberellic acid, cytokinins, and various other factors and stringent conditions to grow these transgenic plantlets in the laboratory conditions initially, which will later be transferred into the field. So here I have expressed, one thing is like, okay, you have isolated a particular gene of your interest, say a toxin which will kill the insects and therefore eliminating insect pests in the crop. So that toxin, the corresponding gene of that toxin has to be cloned and then put it in an expression cassette, that means in a vector, and that vector has to be now inserted or pushed into the plant genome. So how are we pushing it? We are pushing it through two different methods. the most commonly adopted method earlier was agrobacterium based. I will discuss that because it is very prominent method, and the latest technology we have is gene gun. It's like a short gun where DNA is coated with certain metals and pushed into the plant tissue or plant cell. Now you see here, you know, Agrobacterium tumefaciens is again a soil bacterium, all right, which whenever a plant has a wound, that means whenever its tissue is ruptured, Agrobacterium tumefaciens can detect using certain chemicals, and it can infect the wounding site of a plant, and it can form a what is known as crown gall tumor. So what is happening here? Agrobacterium tumefaciens is a bacteria, and this bacteria has a plasmid which is known as Ti plasmid. Now whenever there is a wound, this Ti plasmid is getting integrated into the plant genome, and it is causing a crown gall tumor. Now this is the natural thing which is happening. This is a natural thing which is happening in nature. Now this particular phenomenon has been exploited for the purpose of recombinant DNA technology. How? So you have Agrobacterium tumefaciens, and this is the Ti plasmid. And for every plasmid, you have an origin of replication. That is, the replication starts at this site. And then you have certain vir genes. Vir genes are virulence genes. Now, these are the genes which are responsible for forming crown gall tumor. All right. And then you have a specific portion here. You have a specific portion which consists of auxin producing gene, cytokinin producing gene, opine producing gene, and towards the left you have the left border and you have the right border. Now let us say virulence genes is something which is causing virulence, that means which is infecting the plant. Let us say I have removed this, and I have removed auxin and cytokinin also, because anyways I am giving this auxin and cytokinin in my plant tissue culture. So I have removed this also. I have removed this opine also, which is not required. Now to the left border and the right border, if I insert my gene here, my gene means any gene which I want to insert it into the plant. And this is serving as a vector now. This is serving as a vector now. You see here, this is the left border, right border. I have inserted the target gene here, and you have origin of replications. See, origin of replication is the site at which the replication starts. So if you put another origin of replication here, it can replicate in E. coli. If you put origin of replication of Agrobacterium tumefaciens, it can replicate in Agrobacterium tumefaciens. Now you have a selectable marker. Why you need a selectable marker? We need a selectable marker because once the target gene is inserted, we have to test whether it is inserted at the right place, whether it is inserted or not. So now this is the vector. This is the expression cassette I made. So after this expression cassette, what would I do? I would insert, since we are using the property that, I will put this under Agrobacterium tumefaciens back or E. coli, and I shall give it to the plant. And now what happens is this particular plasmid will get into the plant and it will integrate into the plant genome. All right. So when this integrates into the plant genome, this particular gene will be expressed. But remember, it will not give you crown gall because we have removed the virulence genes. So it is like, you are cutting copying and pasting, in the sense you're cutting certain portions, you're adding certain portions, you're removing certain unwanted portions, and you're making a kind of plasmid which can be grown in bacteria and which can also get into the plant. So you see, initially you start with a lot of data. If you take that total pipeline, initially you start with identifying various events. Identifying various events means where I am inserting my gene, whether I am getting the desired trait, or so. Initially you start with a lot of events, and down the lane, like 5 to 6 years down the lane, you will zero down to one or two events. One or two events means this particular combination of promoter, this particular combination of terminator, and at this site of plant genome, this is now fixed. Now once that is fixed, that means the event is fixed. So now this event you will actually patent, and then you will try to produce it F1 hybrid generation. You will test it for 5 to 10 generations. You will obtain seeds from it, and again you will test the seeds, and these seeds are what are marketed into the market for the farmers. Like for example, in India we have so many companies which sell BT cotton seeds. Now these seeds are again sown into the field, and we expect that it will be we will get a desired property. That means if we are putting a toxin inside a plant which is not itself toxic to the plant, but it will be toxic to the insect or pest, let us say. So in that sense, the crop, you are getting higher yields of a crop, you are using less amount of chemical pesticides, and finally you are also getting a pest free crop. So this is a win-win situation for everyone. Now, after discussing how a GM crop is produced, I will now talk about the two important transgenic crops which we are looking forward in India is BT cotton and BT mustard. Now, this should not be called as BT mustard. We have to call this as GM mustard. So, BT cotton is very well known to all of us, as it is grown in almost like 13 to 14 states, and we are, currently highest exporter of cotton. So this BT cotton is nothing but you have a strain which is known as soil bacterium Bacillus thuringiensis. It has various subspecies, you can see here. And this is a very simple soil bacterium which has two different phases, that is the sporulation phase and the vegetative phase. All right. So during its sporulation phase, it produces a toxin which is in the form of parasporal crystal. Okay. Now this is the parasporal crystal, which when taken up by an insect, okay, when taken up by a pest, now this is produced in the bacteria, that is Bacillus thuringiensis. Now once an insect ingests, how will the insect ingest? Like, for example, I'll take an example of Pectinophora. Pectinophora is nothing but pink bollworm. Or I can take the example of bollworm, that is Spodoptera. These are very, very, very prominent pests across India. Wherever you go, you will have this problem. So if a pest is ingesting this particular toxin, now where is this toxin? This toxin is present in the leaves. This toxin is also present in the leaf buds, or this toxin is also presented in the flower buds. So whenever a leaf or a part of a plant is consumed, cotton plant, now this toxin is expressed using what you say recombinant DNA technology, and which is solubilized into a protoxin. The protoxin is of approximately 250 kilodaltons. Now, once it is solubilized in the gut, the insect gut, if you talk about insect gut, it has an alkaline pH. The pH of the gut of an insect is alkaline, and it also has certain proteases. Now, with the help of this alkaline pH as well as proteases, the protoxin now is getting what do you say, activated into an active toxin. That means a protoxin has to be cleaved by the proteases, and at the pH and at the gut pH, the proteases function optimally and they cleave and to generate an active toxin. Now, what does this toxin do? This toxin will form yeah. Now, what does this toxin do? This toxin, now this portion is your gut. This is the uh, lumen of the gut, and this portion is the lumen of the gut, and this is the microvilli of the gut. And on the microvilli, you have certain receptors. Okay, and these receptors are like now established as cadherins, aminopeptidases, alkaline phosphatases, ABCC transporters. There are certain receptors to which this cryotoxin will bind. Yes, please. Am I audible to you?
Yes, sir.
Sir.
Sir, you are audible, sir.
All right.
Yes, sir.
Yes, sir.
All right. Thank you. So once this cryotoxin binds to the receptors, there is an oligomerization. That means various such cryotoxins will form as a group, 5 or 6 oligomers, and that particular toxin, now you see one monomeric unit is forming a kind of a channel, a pore. 4 or 5 such units may form a pore inside this gut. So once there is a pore formation in the gut, once there is a pore formation in the gut, all the gut contents, this is the gut lumen, so all the gut contents would be leaking out, and there is also an efflux of ATP, and within a day or two, the insect, because of desiccation, because of ionic imbalance, and because of this efflux of, because this is the porine toxin channel which has been created, because of this, now the pest will die. So this is how when you talk about the cryotoxin from the bacterial Bacillus thuringiensis, you have put it in a vector again, which is a vehicle. The vector looks something like this. I'm not going into the technicalities of it because I wanted to give you an overview of the whole thing. So you have this vector where you will insert certain cryotoxins. Now, the 2 cryotoxins which are approved in India are Cry1AC and Cry2AB. There are so many cryotoxins, but these 2 are approved. Now you put these 2 toxins, the genes of these 2 toxins, under the control of a promoter, under the control of a terminator, and you have various selectable markers, and you put it in an insert. Now this insert, using again gene gun technology or Agrobacterium tumefaciens, you can insert it into the plant genome.